Hi, thanks for joining me. I'm Dr. Michael Fossel. I'd like to talk about something that's very extraordinary. It's something without an historical precedent. It is the prospects for being able to cure the age-related dementias and specifically Alzheimer's disease as well as others. Now, it involves a novel approach and a deeper understanding of the disease itself and the aging process itself. The result is an innovative way of looking at the disease and an innovative way of caring for the disease. Too often, most of us, understandably, focus on just treating the signs and symptoms. That is, somebody has Alzheimer's disease and we try to make them as comfortable as we can over the last few years of life. But I think most of us would wish to go deeper, would wish to actually intervene and find a way to actually cure the disease itself rather than just the symptoms. So with that in mind, let me suggest that sometimes what we do is we hunt the rabbits rather than the tigers and dragons. And the rabbits in this case are the signs and symptoms, rather than actually being able to go for something truly remarkable, which is to cure the disease itself. And I think we can do that, but only if we actually admit that we don't understand it. As Stephen Hawking says, the problem is not that we don't know, but that we don't, that we think we do know, and that gets in our way. And the same has certainly been true of Alzheimer's disease, where we tend to think it just happens and we understand it. We don't. Another way of looking at it is this quote from Einstein. Too many people know things, but they don't understand things. And a classic example would be to go back 500 years to the Vatican astronomers, who were enormously knowledgeable about planetary positions, but they had no understanding of orbital mechanics. And that was really left to Kepler and Copernicus and Galileo, who took us a lot further than just knowledge. They took us to understanding. Well, much the same thing is true when we look at age-related diseases, for example, Alzheimer's. We have an enormous body of knowledge out there. We know a lot about beta amyloid and tau tangles, microglial activation, and mitochondrial dysfunction. But what we lack is an understanding of the process that ties all of these things together and makes it into a single body and allows us to intervene effectively. I first became aware of this sort of problem about 45 years ago on this campus. This is Stanford University in California, where I did my PhD in neurobiology and then was teaching neurobiology in the medical school while I did my MD. And what I found is that most people thought they understood aging. They thought it was just entropy, it's wear and tear, things fall apart, and what do you expect? And I found it was a great deal more than that, a great deal more complex. And it resulted in me giving the first talk ever in human history on the prospects for reversing human aging here at the NIH in Bethesda 25 years ago this year. And what I suggested was that if we truly could understand the fundamentals of aging at the cellular, the genetic, and the epigenetic level, then the prospects were there for us to be able to intervene effectively in age-related diseases and actually cure and prevent age-related diseases rather than just treat sort of the signs and the symptoms of the diseases. Well, with regard to aging in the nervous system, for example, the dementias and specifically Alzheimer's, what you find is that these two quotes have all too much truth to them. The researchers will tell you that everything works in mice, nothing works in humans, and as we'll see, it's not just mice, but it is true they don't work well in humans. And investors will tell you that Alzheimer's is the graveyard of companies, and that's true of not just small biotechs, but major big pharmaceutical firms like Biogen. So when I say it's not just mice, we also find that the same is true of dogs and cats and baboons and rhesus monkeys and humans. As we try to go from animal study to humans, you get these tantalizing bits that suggest that maybe some of these things might work in animals, but when you try to take them to human trials, they simply don't work. And of course, the question is why? And it's also true that nothing really does work in humans. These are some of the facts. These are taken from clinicaltrials.gov, and what I've done is listed all of the interventional trials, for example, for Alzheimer's disease. There are more than 2,200 of them. And by global consensus, all of them, without exception, including things like Biogen, Eisai's, and aducanumab study, have failed to alter the progress of the disease. So this is where we stand with regard to, for example, current monoclonal antibody approaches to treating Alzheimer's, whether it's beta amyloid or tau tangles. What you find is that there's been one compound that has been released by the FDA for clinical use in January of 2021, that is aducanumab. But no matter how we look at it, even the best of those gets this sort of result. This is the data from aducanumab for different dosing levels, and as you see, the third highest level takes an odd course. But what you'll notice is that, well, there's this temporal step-off, a delay of the onset of symptoms for as much as 6 to 8 months at best, giving you more time in the nursing home. None of these affects the ultimate course of the disease. And all the patients have a disease course that parallels that of placebo, which is they not only go downhill, but if you treat them, they have about a 40% risk of adverse reactions. Scarcely what we'd want. What we would like to see is this. We'd like to see that we have an ability to stabilize the disease, or even better, to show that we can improve the cognitive status, not have any adverse reactions. But why haven't we done this? And the answer really is that we've been digging in the wrong place. We have been trying to target downstream outcomes, that is, signs and symptoms of Alzheimer's, for example, beta amyloid plaque and tau tangles and so forth. But what we have not done is ask ourselves why these things work. So the consequence is that we've been digging in the wrong place. And when you're doing that, it really doesn't matter how much money you put into it. I mean, the NIH budget for 2021 is estimated at almost $3 billion. The Biogen Eisai costs of developing aducanumab were about $1 billion. And when you digging in the wrong place, it doesn't matter how much money you put in, how big your excavator, how well trained your personnel, how many people you have digging. It doesn't help if you're digging in the wrong place. So, in consequence, about now almost 2 years ago, in 2020 though, I put out this article that has attracted considerable attention globally, with more than 1,000 reprint requests and invitations to give talks and run conferences like this talk. And what I'm really saying is that if we look carefully, we can understand how to unify and understand the systems view of the age-related dementias and neurodegenerative disease by starting to look at cellular aging. So when I say unified systems model, let me be a little more specific. When I talk about unified, what I'm really saying is we need to be able to encompass everything we know about Alzheimer's disease, for example. So we know that certain genes have increased your risk of Alzheimer's, but why? We know that exposure to certain toxins like paraquat and dioxin increase your risk, but why? We know the same is true of head trauma, hypertension, hyperglycemia, exposure to a number of infectious agents, for example, fungal infections and viral encephalitides, but why? We also know that downstream, you get beta amyloid plaque, or you get alpha-synuclein aggregates in Parkinson's or tau tangles, but why? What we lack is an understanding of the central mechanisms, these unified fundamental processes that underlie age-related neurodegenerative diseases. That's what we need to unify. Looking at it clinically, let's look at a number of the common syndromes we see: Alzheimer's, Parkinson's, frontotemporal dementia, and vascular dementia. What you see is that while these are all too real, they also overlap. So what you find is that a number of patients who have Alzheimer's will have Parkinson's-like symptoms, or Parkinson's patients will have some Alzheimer's-like symptoms, and many of these patients at autopsy will show vascular dementias as well. And that's true no matter where you look in the alphabet soup of age-related dementias. What you find is that while there really are these syndromes, many patients have atypical presentations or mixed presentations, leading many of us to suspect over the past few decades that what we're dealing with is an age-related neurodegenerative disease that has a fundamental shared mechanism for all of these different age-related dementias. The question is, what is that mechanism? Now, when I say a unified systems model, let me be more specific, because it's a subtle but critical distinction to make. When I talk about a system, let's look at an engineering system. In the lower right, you're looking at the fan blade of, in this case, a Trent 900 turbofan that powers that Pearl 15 Rolls-Royce jet engine in the upper right. That is the main power source for, in this case, the Airbus 380. You're looking at the cockpit in the lower left. And when that fan blade fails, it's not a failure of just the fan blade. It's a failure of the fan blade as part of the system. That is, as part of the gestalt, part of the whole process. That is, it's in a rapidly rotating turbofan in the rapidly rotating and at a high temperature Pearl 15 jet engine in an Airbus going hundreds of miles an hour at 35,000 feet. So the question isn't just about the component. The question is, how does it operate in the system? What's going on in the entire system that makes that fan blade fail? And the same is true of medicine. If you think about, for example, COVID infections, these are typical signs and symptoms of a COVID Delta infection, but they're no more than biomarkers. They're not the disease itself, they're not the system. The system is not this series of symptoms, no matter how carefully you measure it, and you should, but it involves a process, a system at a much deeper level, that is a viral infection and the human immune system and a number of other organ systems. And the same is true of aging. These are macroscopic biomarkers, hallmarks, components of aging, but they're not aging. They're simply downstream outcomes The same is true if we look at the microscopic level. None of these things, telomere shortening, acetylation, methylation, declining ATP ratios, none of these things are aging. They're part of the process, they're part of the system, but they are simply biomarkers or components of aging, not the dynamic system. And when we're thinking about the dementias, the same is true. We can look at all of these things, blood-brain barrier failure, alpha-synuclein aggregates, lymphatic changes, and so on. But these by themselves are not dementia. They are components, biomarkers, hallmarks, downstream outcomes, upstream risk factors. They're not the process itself, the system. And that's what we need to understand. The same is true when we look at the genetic level, at the finest possible level. We know more than 200 genes that may mostly increase, occasionally decrease your risk of a number of the dementias, for example, ApoE4 alleles, but those aren't dementia. They are part of it. They need to be understood. We need to explain how they work, but they're not the dynamic system involved in dementia. Until we understand that, we can't go further and cure the disease. And in fact, as Len Havlik said, most of us would agree that aging is the single major risk factor for age-related diseases, but then we entirely ignore the aging process and we focus on the outcome. That is the histology, the physiology, the proteome, the genetics, without looking at the aging process. Now, it was Len Havlik who 60 years ago this year first described cellular aging. And that's where we should be looking if we're going to understand aging itself. So if we're going to be able to intervene effectively in age-related diseases, what we need to do is understand the aging process and ask this simple question, which is, what is aging? Not simply entropy, but what's really going on. If we look at a number of different species, we think we begin to get some clues. For example, humans have a lifespan of about 3/4 of a century. Gorillas just short of a half a century. This is Coco, the gorilla I babysat for a year at Stanford when she spoke sign language better than I do. Typical domestic animals may live 15 to 20 years, depending on the species and breed. Typical rodents, laboratory rodents especially, 2 or 3 years, depending on the species and breed. And it's very tempting to say that aging is just entropy until you realize that all of these animals have different lifespans. It's then very tempting to say, well, the reason they have different lifespans is they're genetically different, which doesn't explain a thing. Mice, for example, have about 50% or 75% of identical protein expressing genes to you and I, but that other 25% is different, and then there are regulatory genes. So we know that it has something to do with genes, but it doesn't tell us what's going on. And the same is true even when you look at single species and single genes. So for example, these are 30 Hutchinson Gopher progeric children. And 20 years ago, I was gathering together from all over the world, and this particular year-old Bill Sample in the background with his hands up, and I'd taken them for a tour of the White House in places you are not allowed to go. And these children have an average lifespan of 12.7 years. Every one of them is dead right now. They die overwhelmingly of cardiovascular diseases like strokes and heart attacks, but they also have baldness, osteoarthritis, osteoporosis, and other age-related diseases. Well, we think we, quote, know why. That is, they have a lamin A gene that's abnormal. But notice that that doesn't explain how it works. What we've said is there's some association, some correlation with the gene without understanding causation and the systems process involved. Furthermore, aging is not ubiquitous. Every cell in your body has demonstrated aging over the past couple of decades, but you inherited a cell from your mother, who got it in turn from her mother, who got it in turn from her mother, and that cell line can be traced back almost 4.5 billion years. But when you inherited it, it was fully functional, had no evidence of aging damage. And yet, in a matter of decades, your somatic cells show aging damage. So why does aging not occur in some cases and does in others? It's tempting for some people to think it has something to do with mitochondria, but you run into the same problem. Every mitochondria in your body was inherited as a fully functional mitochondria that you got from your mother, got it from her mother, and that line goes back in the eukaryotic cell line for something short of 2 billion years, depending on who's making the estimates. So you inherited a more than a billion-year-old cell or mitochondrial line, and it was fully functional and showed no aging changes, and yet in a matter of decades, most of your mitochondria show age-related changes. Why do mitochondrial dysfunctions not occur for a billion years, and then they occur in a matter of decades? Why the difference? Well, part of the answer is to understand the difference between entropy and maintenance. Aging is purely not entropy. It is entropy in the face of failing maintenance. So for example, the germ cell line managed to maintain itself for that billion years, as do the mitochondria you inherited. But when I look at the cells in the mitochondria in that 80-year-old man's hand on the left there in the wheelchair, you find that no matter what cells I look at, keratinocytes, fibroblasts, vascular endothelial cells, microglia, these cells show subtle deficits in maintenance. You might even say that the problem is not being able to keep it running in the same place. This is a quote from Through the Looking Glass by Lewis Carroll, where the Red Queen says you must run as fast as we can to just stay in one place. That's what your cells do in the face of entropic change or wear and tear. They maintain themselves rapidly in the face of that, and in consequence, they manage to keep going. Looking at that from the molecular level, you can look at any molecular pool in the body that you want to. Beta amyloid, collagen, and elastin between the cells in my face. You can vascular endothelial cells, renal podocytes, pulmonary pneumocytes, no matter where you look. What you find is that any particular molecular pool you look at within those cells in any particular pool, you find that in young cells, they are turning over things very quickly. That is, recycling molecules. So they produce a molecule, they break it down, they produce it, they break it down. And in consequence, you find that the damaged molecules, signified here by the little red star, are minimal. So in young cells, there's very little actual damage because it doesn't accrue because of the rapid recycling rate. But as you get older, that rate of turnover slows down as a result of changes in gene expression. And the result is you get this gradual increase in the percentage of damaged molecules, which leads to damaged cells, which leads to age-related diseases. In fact, you can look at this mathematically. This is taken from my Oxford University Press textbook on aging. What it really says here is that if you have a rapid rate of turnover in young cells, you have very little damage. If you have a slower rate of turnover, you'll have a great deal more damage and therefore dysfunctional cells. Well, let's take that down now to the central nervous system and look at one example, which is Alzheimer's, and one example within Alzheimer's, which is beta amyloid proteins. And what you find is if you measure beta amyloid turnover in microglia in human and other animal brains, you find that in young microglia, that turnover is very rapid, and in consequence, there's very little residual damage. But as we get older, the turnover rate slows down, and the result is you begin to get these beta amyloid microaggregates. You might reasonably ask, what about genetic risk factors? And the answer here is, if you have, for example, low-risk alleles like ApoE2 alleles, you make a form of beta amyloid that's not very sticky. So even as you begin to get older and your turnover slows down, you're not accumulating much in the way of beta amyloid microaggregates. On the other hand, if you are at higher risk and you have two ApoE4 alleles, you make a stickier form of beta amyloid. So even at a younger age, even at your 20s and 30s, you're beginning to accumulate more in the way of beta amyloid microaggregates because of this gradual early slowing of molecular turnover, which occurs within your brain. But as we get older, in our 80s and 90s, all of us begin to accumulate these beta amyloid plaques. It's just a matter of your genetic risk factors and your age. Now, the same is true when we look at the outcome, synaptic loss, cellular dysfunction, the clinical dementias. All of this, including other changes, tau proteins, mitochondrial changes, inflammatory changes, all of these changes can be ascribed to changes in gene expression modulated by cell senescence and telomere shortening. And the answer is all of this leads to disease in all of these different venues. So, for example, if we look at mitochondria, now most of the aerobic enzymes in mitochondria derive from the nucleus. And in older cells, you find that the rate of turnover of those mitochondrial enzymes is slower, so that there's more damage accumulating in the enzymes, for example, cytochrome C oxidase. The result of that is you get less and less in the way of power production, that is ATP, and more in the way of free radical production, that is reactive oxygen species. So you're becoming less efficient. Not only that, but the lipid membranes that safeguard the rest of your cytoplasm and your nucleus from these reactive oxygen species are also being turned over more slowly. The lipid molecules are being turned over more slowly, so there's more oxidative damage, so they become more porous, and so you make more reactive oxygen species and you leak them more. Also, the scavenging molecules like catalase and superoxide dismutase are being turned over more slowly. So once those reactive oxygen species leak from the mitochondria, they're not being caught as quickly. And so when there's damage to other molecules, the recycling rate has also gone down. So for 4 separate reasons, independent reasons, the increased reactive oxygen species production, the increased porosity, the decrease in scavenging ability, the decrease in recycling molecules, you're getting increased cellular damage because of mitochondrial dysfunction, which can all be traced back to changes in genetic expression within the nucleus, which are modulated by telomere shortening. The same is true of DNA damage. If you look at the 4 major families of DNA repair enzymes, what you find is that all four show these subtle deficits in the rate of recycling. So for independent reasons, each of them are turned down, and the result is an exponential decrease in the rate of DNA damage repair. So every cell in your body has shown some DNA damage in the past 20 minutes since I've been talking. But the result of that has been almost flawlessly repaired, but more slowly as you're going to get older, which is why there's an exponential increase in cancer with age. It's not simply a matter of increased exposure or lifetime exposure to carcinogens. It's also a lifetime decrease in your ability to repair DNA. So overall, what we're looking at is this. We have a number of upstream factors and dozens more that have an impact on the rate of cell aging and the type of cell aging that occurs. And then we have a number of downstream findings, hallmarks, biomarkers, signs and symptoms of aging, outcomes of aging in a clinical sense. But all of these work together. And when we try to intervene upstream, we find that we can affect some of these things. For example, we can try to lower your risk of head trauma or control your hypertension or your hyperglycemia. But none of those actually stop age-related disease like Alzheimer's. They decrease the likelihood or they postpone the onset of, but they don't stop it. The same is true of the downstream factors. So for example, when we try to intervene at the level of beta amyloid plaque, we find that things like anakinumab simply don't do the job. Can we do better? Can we intervene in the fundamental process of aging itself in cells? And there the answer, oddly enough, is yes. We not only can, but we have. This is a paper in Science 23 years ago by Andrea Bodnar, group in California. And what they showed is that you can actually reverse cellular aging in human cells in the laboratory. You can reverse the pattern of gene expression, the rate of turnover, the physiology, the histology, the morphology of these cells. Not only can you do it in cells, but as it turns out, you can do it in tissues. This is another classic paper from Walter Funk's group 21 years ago, and what he showed is that you can literally grow young human skin cell or young human skin tissue from old human cells as long as you reset telomere lengths and therefore reset the pattern of gene expression. Well, as it turns out, the same thing is true of bone cells, and the same thing is true of vascular endothelial cells, suggesting that we could find a much more effective way of intervening in age-related cardiovascular disease. Now, the question really is, can we do something about this in organisms, that is, in animals like human beings? And the first study that looked at that in any reasonable way was about a decade ago by a group at Harvard, under Pinto's group. But what they did was they altered the germ cell lineage, which is not something I can really do effectively in human trials. But the second study that looked at this was this article put out 9 years ago by Maria Blasco's group in Madrid. She's the director of CNIO there, a big cancer research institute. And the De Jesus paper showed that you can not only increase the lifespan, but reset behavioral patterns of old animals to those of young animals, as well as a lot of other physiologic measures like bone mineral density, subcutaneous fat, and immune function, and others, simply by using telomerase to reset cell aging and therefore tissue aging in these organisms. So let me summarize what we know about cell aging. It is the central mechanism and it's the optimal point of intervention. We know it's a platform therapy. We can use it to approach central nervous system cells like microglia and therefore treat Alzheimer's. We can use it for vascular cells, chondrocytes, for example, in joints, osteocytes in bones, renal podocytes for chronic renal failure, leukocytes for immune senescence, skin cells for wrinkles and other age-related skin changes. Furthermore, we know that this model encompasses all known upstream risk factors. It explains why they are risk factors. It encompasses all the downstream biomarkers and explains why those things occur. And it explains clinical heterogeneity. Why do I get Alzheimer's, you get Parkinson's, and someone else gets frontotemporal dementia? It also explains the heterogeneity between species. Furthermore, it is consistent with all the known data that we've got, both research data and clinical data, and it's predictively valid. This model not only explains, but predicts ahead of time the results of the Eli Lilly solanezumab trial, the Biogen Eisai trial with anakinumab. Furthermore, this offers us a novel target that is technically within our reach. We can do this. We have the mechanisms to treat human beings with this, and it's demonstrated that it works in vitro, ex vivo, and even in vivo in animals. For clinical trials, what we're focusing on is credibility. We want to make sure that we do this rigorously, we do this correctly. We will also obviously be looking at efficacy, safety, the cost, and timing of this. So let me ask two basic questions. First question is, can we reverse cell aging at the cell and tissue levels? The answer is yes. That has already been done. been done multiple times in multiple cases, both in vitro and in vivo. More important question is, can we reverse cell aging in human clinical trials? And there the answer is, we probably can. And we're going to show that we can do this as soon as we reasonably can do it. I'd like to thank you all for joining me in this talk. If you have any questions, I hope they'll get back to me. As I say, I think we can do something that has never been done before in human history, but it requires that we truly understand the aging process at the most fundamental levels. Thank you again.